I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I'm your host, Jesse Lawler, excited to bring you the 96th episode in this podcast dedicated to the ongoing improvement of your own brain by any and all means at your disposal. This week, I am happy to bring you another installment in the every now and then Know Your Neurotransmitters episode series. This is going to be our third. We started off with dopamine and then did GABA, and now we are moving on to Noradrenaline. Not to be confused with adrenaline, which is a related but different chemical. We're going to learn about how those two relate to one another. We're going to be talking with Dr. Clare Stanford, who shares a last name with one famous university but works at another. She is at the University College London over in the UK and has possibly the most charming British accent that we've ever had on this show, but I'll leave that for you to judge. If you hang around until the very end of the episode, I'm going to tell you about a kerfluffle going on in the world of Chinese biotech and a really interesting discovery that's at the root of that kerfluffle. And yes, I might continue to use the word kerfluffle just because it's fun to say. That will be in the Ruthless Listener Retention gimmick, but as usual, let's kick things off with This Week in Neuroscience.
Smart Drug Smarts. This week in neuroscience.
So if you've got young kids, you might have noticed that they are not as proficient as adults at holding a lot of things in their memory at once. They get distracted. Remembering more than a few things at a time is possibly not going to happen. And while parents have known this since the dawn of time, we haven't really known why, exactly what's going on, and why the working memory of adults seems to be a lot better than the working memory of young children. But researchers at Northwestern University, working together with colleagues at Boston Children's Hospital, have been looking into this question, says Maria Karatanova. Previous work pointed to the fact that children use their prefrontal cortex less than adults, but we wondered why. And if they're not really using their prefrontal cortex, but they are remembering things, what parts of the brain are they using? So in this new study, researchers got both children and adults, children were aged 5 to 8, to line an fMRI scanner and look at different colored dots on a computer screen. Every few seconds, the dots would switch around and the people would need to identify whether the new dot that they saw was the same or a different color than the previous dots. Adults could pretty reliably do this for up to 4 dots, but for kids, it was really hard. The kids maxed out at about just 1 to 2 colors, and they were using their prefrontal cortex just as much as the adults who were doing 4 colors. But the prefrontal cortex was not the whole story. Children also just activated some other areas less, like the parietal cortex. Their activation in these areas increased as the task became more complicated, but they never got to adult levels. As for how to use any of this information in practice, Karatanova says it's important just to recognize that young children can't hold as many things in their head at one time. And the next time that your 6-year-old forgets the 3 things you told him, realize that it's better to only get him to remember 1 or 2 things effectively than to try for 3 and lose the whole kit and caboodle.
Smart Drug Smarts, the podcast where smart people talk about smart drugs.
I would like to thank the several people that reached out to me over the course of the past week on Twitter and expressed your condolences for my having run out of mitogen. And yes, that problem has now been solved. I'm happy to say that my medicine cabinet is fully restocked. Also got some vitamin D3 and a couple other things over the course of this past week. So no longer shall I be palming a shabby half handful of pills in the morning. It's back to its full handful proportions. I'm getting a bit behind on my emails again. So if anybody's emailed me anything in the past week and I haven't responded, apologies. I will be trying to clear that backlog soon. I do know that the lovely and talented Rhiannon Rowe sent out an email newsletter to the Smart Drug Smarts mailing list. If you're not on that mailing list, I would encourage you to take a visit to smartdrugsmarts.com and quickly and painlessly enter your email address in there so we can keep you up to date on everything that we've got going on, which, if my sources are correct, might soon include a very interesting article based on an interview that I did with Dr. Elizabeth Loftus, who is an expert in false memories and how we can intentionally or unintentionally construct false but extremely convincing memories, and how this can be advantageous in some cases and extremely bad in other cases. This was an interview that I did a few days ago. It wound up being a really bad telephone connection. Unfortunately, it's not really audio that we can use for the podcast, but the information was great, so I think that's going to wind up on the website. Anyway, stay apprised of all that stuff by jumping on the smartdrugsmarts.com mailing list if you're not there already. And finally, I would like to thank you, our Smart Drug Smarts listeners, for having made this past month, September, which just came to an end, our biggest month ever for the 5th month in a row. It's really great to find out that new people are finding out about the podcast, and I think it's fair to say that a giant proportion of that is coming from word of mouth and the digital equivalent of word of mouth. So thanks to all of you that are sort of banging the drum on our behalf. We are in the process of putting together an Adderall episode very similar to the Jesse's self-experiment with methylphenidate episode that we did a month or 2 ago. I'm probably not going to be doing Adderall myself, but we do have somebody who's going to come on and talk extensively about his first-person experiences with it, and we're looking to get some other people to come in for sort of micro interviews just so we can get a variety of people's experiences with that that drug. If you are a past or present Adderall user and you feel like sounding off on your thoughts, drop an email to info@smartdrugsmarts.com and just say, hey, this is something I have some experience in. I'd be interested in doing a micro interview, and we'll get back in touch with you. And for those of you who are skittish about things like Adderall but do like over-the-counter supplements and cognitive enhancers, please do consider taking a spin over to axonlabs.io on the web. Axon Labs is the retail wing of what we do over here at Smart Drug Smarts, where we've got our two stacks, Nexus and Mitogen. Nexus is our cognitively focused stack based around aniracetam, CDP-choline, pycnogenol, and phosphatidylserine. And Mitogen is a mitochondrial stack, which also has quite a bit of a cognitive kick, thanks primarily to the cell-buthiamine, which is one of its major components. Both of those can be found over at axonlabs.io and are available for single purchase and ongoing subscriptions.
Smart Drug Smarts.
So we are about to be talking with Dr. Clare Stanford from the University College London. She has a doctorate in physiology from Oxford University. She's also a past president of the British Association for Psychopharmacology and is an editor of several international journals, including the British Journal of Pharmacology, the Journal of Psychopharmacology, and the International Journal of Neuropsychopharmacology. Today we're going to be talking specifically about noradrenaline, which you will also hear referred to as norepinephrine. These are exactly the same chemical, so we're going to be calling it noradrenaline today, but don't get confused. This is just one of these British-American difference things. Epinephrine is adrenaline, norepinephrine is noradrenaline. It's a little bit annoying, but it's not that confusing. So you can consider this the noradrenaline or the norepinephrine episode at your pleasure. You'll notice a couple minutes into this interview, there's a little patch where the audio gets rough, but then it pops right back after about 10 seconds, so don't get scared off about that. Noradrenaline winds up being a really interesting neuromodulator. I started off by calling it a neurotransmitter, but Dr. Stanford sets me straight within this interview, so listen up for that. But here we go with Dr. Clare Stanford.
So I always look at noradrenaline as, if we had to give it a label, I would regard it as the transmitter that makes sure we can look after ourselves. We have to think things are worth doing or we wouldn't do anything. And that may be what's partly going wrong in depression, that people have a motivation problem where they just can't even face getting out of bed. It's just not worth it. And it's a combination of interacting with noradrenaline and dopamine that actually gets us out of bed. Dopamine makes us a tenth of the life that we're going to live, and noradrenaline enables us to do it. Noradrenaline or adrenaline comes from the same synthetic pathway, and so does dopamine actually. They all share that route, and it starts off with an amino acid called tyrosine, which is taken in the diet, and tyrosine is converted into L-DOPA, and L-DOPA is well known for its efficacy in treating Parkinson's disease. L-DOPA is then converted into three chemicals which are all known as catecholamines, and the first one is dopamine, the first in the pathway. And then dopamine is converted by beta hydroxylation into noradrenaline. And then noradrenaline is converted into adrenaline by an enzyme which is given the acronym PNMT, phenylethanolamine and methyltransferase. And what makes the difference is whether the neurons actually express those enzymes or not. So a dopaminergic neuron will have the enzymes required to produce dopamine, but they will lack the enzyme dopamine beta hydroxylase, which is needed to convert dopamine into noradrenaline, and noradrenergic neurons lack the enzyme PNMT, which is needed to convert noradrenaline into adrenaline. So the phenotype of the neuron is really determined by what enzymes they express.
So tyrosine within our diet, then that amino acid is sort of at the root of all of this.
Absolutely, yes. The tyrosine is fundamental, and that's converted into L-DOPA by an enzyme called tyrosine hydroxylase, and that's a very important enzyme because it's the rate-limiting step in the whole pathway. So the activity of tyrosine hydroxylase will govern how much dopamine, noradrenaline, or adrenaline we're able to produce in all those neurons. It's the bottleneck. That said, its affinity for tyrosine is extremely high, and normally that enzyme is 80%, if not more, saturated with a substrate. And so as soon as it binds any tyrosine, it whips it through to dopamine straight away. If you want to increase the activity of tyrosine, then there are various steps which can increase its affinity for tyrosine even more or increase the maximal activity of the enzyme. But tyrosine availability is very, very rarely a limiting factor in dopamine, noradrenaline, adrenaline synthesis, because that enzyme has such a high affinity that it just grabs everything it can get hold of.
So it's never a situation where somebody goes to their doctor and their doctor says you need to get more tyrosine in your diet?
Not typically. If we ever became tyrosine deficient, I think we probably wouldn't be able to get to our doctors. We'd be so moribund. But there is one situation which is extreme interest to psychopharmacologists, in that you can actually manipulate the diet to produce a tyrosine-free diet and put lots of other amino acids which would compete with tyrosine for uptake into the brain. And in those situations, it is possible to reduce the supply of tyrosine. And we find that patients who are in remission from depression, who have responded to antidepressants that target neurons in the brain that release noradrenaline, then a tyrosine deficient diet can actually cause a resurgence of depression. So under normal circumstances, tyrosine availability would never be rate limiting, but it may be that sufficient tyrosine is required for antidepressants of that type of antidepressant to continue to work. So tyrosine may be essential for antidepression, but doesn't seem to have any particular impact on normal existence. That distinguishes the pathway for noradrenaline from serotonin, for instance, which in many ways are analogous. But the amino acid that starts off the serotonin pathway is called tryptophan. And tryptophan availability really can be rate limiting in normal life. And more importantly, if you supplement with tryptophan in the diet, then you can end up producing more serotonin. The neurons store more serotonin, they release more serotonin. And that can have quite profound effects on mood and behavior. In fact, tryptophan supplements were at one time considered as a way of treating depression. But tyrosine really isn't like that.
Gotcha. So within the body then, noradrenaline is sort of kept much more constant. It doesn't float up and down, fluctuate up and down over the course of a day or a week or a mood or something like that, the way that serotonin might.
That's absolutely right. Noradrenaline in the body is really found in three places. It's found in nerves that we call sympathetic nerves. They're the nerves in the rest of the body that supply the vital organs, the blood vessels, heart, salivary glands, pupils, that sort of thing. It's also produced in huge quantities in the adrenal gland. People associate the adrenal gland with releasing lots of adrenaline, but actually most of the noradrenaline we find in plasma comes out of that gland too. And there are also clearly topographically arranged noradrenergic neurons in the brain. And all those neurons seem to go to a great deal of trouble to maintain the stores. They're remarkably constant. You can measure them from day to day, month to month. And for each particular organ or brain region, the neurons supplying that organ or brain region have remarkably constant stores of noradrenaline. And we now know quite a lot about the mechanisms to ensure that there's a steady state, so that if the neurons for any reason release more noradrenaline, if they have a burst of activity, then that burst of activity at the same time triggers mechanisms which activate the synthetic pathway. So under normal circumstances, the rate at which we synthesize noradrenaline pretty much balances the rate at which we release noradrenaline. That can break down during prolonged periods of profound stress, but these would be the sorts of stress that are really physiologically demanding. You find yourself on the side of a mountain overnight or something like that, then we might find that noradrenaline stores become a bit depleted. But normally it's exquisitely defined and regulated to make sure that the neurons are ready and waiting to respond to a demand in their activity.
If it exists in such constant supply within the body, what is it signaling?
People talk about levels of transmitter in the brain, and actually that doesn't mean anything at all. There are two ways of looking at levels in the brain. One is the size of the store, and the noradrenaline, indeed dopamine, serotonin, and the rest are all exactly the same, is stored very precisely in big vesicles in the nerve terminals. And those vesicles, they're like little balloons, and they store the noradrenaline, and they're ready to release it. They squirt it outside the neuron when it's needed. And the trigger for that is a nerve impulse reaching the terminals. And these vesicles merge with the nerve terminal and extrude their contents. Now, the rate at which that happens will depend on the frequency of nerve impulses reaching the terminals. So the higher the neuronal activation, the higher the rate of frequency of nerve impulses, the more vesicles will release their transmitter stores, and the more noradrenaline there will be in the extracellular space to hit receptors on the target organs, and that will trigger a response or modulate a response in the next cell in the chain. So when people talk about levels, I always ask, well, what do they mean? Do they mean the amount of transmitter in the storage vesicles just sitting there, not doing anything, but ready and waiting? Or are they talking about the extracellular transmitter, which is really getting busy and bouncing off receptors and triggering responses and so forth?
Now, I noticed just a minute ago, you said trigger and then you stepped back and said actually modulate. That sounded like a meaningful distinction. Can you tell us a little bit about what was going through your mind there and why that distinction exists?
Very sharp of you. I was being very sloppy. I was using sloppy terminology. What we understand by trigger is basically an on-off switch. So a neurotransmitter will float in the extracellular fluid till it hits the receptor. And if it triggers a response, it has flicked the switch and is activated, or it could inhibit in certain circumstances, the response in the target neuron. Now that has to be an ultra-fast response. We want to make sure that every time an instruction is sent from one neuron to another, it's eliminated as quickly as possible. We don't want an event that happened to us on our way to work taking an hour to wear off. We need to be ready to respond instantly to changes in our environment. So fast neurotransmitters include things like the amino acids, which activate receptors, which open pores and flip across from one side of the membrane to another. They're super fast on-off switches. Monoamines actually aren't like that. They're strictly what we should call a neuromodulator. I mean, they're called neurotransmitters, but strictly they're neuromodulators. And what they do is they don't trigger a response. They refine the response. They modulate it. They're like dimmer switches or amplifiers. So when they hit their receptor, that combination triggers or changes the configuration of the receptor, which then interacts with an enzyme inside the cell. It's more like at the onset of winter, if we live outdoors, like most animals do, they begin to feel a bit chilly. That's the physiological stress. They trigger the stimulus, which means in about a week's time, they'll be ready and waiting to produce more noradrenaline in the nerve terminals, which is where it's released. And that's very important because noradrenaline is responsible for increasing basal metabolic rate. It talks to adipose tissue, talks to brown adipose tissue, which produces heat. It's a non-shivering thermogenesis. So there are short and long-term adaptations going on where the noradrenergic neurons can fine-tune their activity according to whatever's going on in the environment. And then, of course, if we're asleep, they switch off altogether. In the brain, if we're not active, then we don't need to have a raised heart rate or conserve blood pressure and so forth. And they buck up just before we're about to wake up. So noradrenergic neurons are needed to enable awakening. So, sleep is an active process. We have to switch on sleep and switch off arousal. And noradrenergic neurons do the opposite. They switch off sleep and switch on arousal.
That sounds a little like how the body spikes in cortisol maybe about an hour before people wake up in the morning typically. Is there any relation there?
Yes, indeed. The neurons in the brain that release noradrenaline, they project down the spinal cord and they do couple with the adrenal gland. The adrenal gland is a bit like a sports ball. It's got a core and a shell, and the shell releases cortisol, corticosterone, and the core releases adrenaline and noradrenaline, and they talk to each other, and they talk to the neurons in the brain, which tell us whether we're aroused or not. So indeed, those two systems are functionally very, very highly coupled. And when we're stressed, of course, we not only release lots of adrenaline, which everybody talks about the adrenaline rush, but we release cortisol at the same time. The signal is the same.
Now, you mentioned adrenaline there. We have these very, very similar names, and I think probably 99% of the audience, myself included, tends to think of noradrenaline and adrenaline kind of as a package deal, or at least they get muddied together in our heads because the names are so similar. Can you kind of splice that for us and tell us how we should be thinking of one versus the other?
Well, they do have a lot of actions in common. They interact with very similar receptors, but they have very different affinities to the receptors. So some receptors have a very high affinity for adrenaline. The classification of these receptors is really historic. And Raymond Alquist, who first discovered the first two receptors for noradrenaline back in about 1949, said he called them alpha and beta because he couldn't think of anything else to call them. We now know that both alpha and beta have lots of subgroups of receptors, but alpha and beta illustrates the point. So alpha receptors have a very high affinity for adrenaline, but a much lower affinity for noradrenaline, whereas drugs that only bind to beta receptors have very important clinical indications. Drugs that block beta receptors, for instance, are probably more widely used than any other type of drug to treat high blood pressure because the heart is loaded with beta receptors. The type of beta receptors you find in the bronchi are slightly different from those you find in the heart.
The bronchi are the branches of the lungs?
That's right. They're the tubes which constrict in asthma. And drugs that block beta receptors in those tubes are used as anti-asthmatics. They have a slight overlap. So people who take drugs like salbutamol to treat their asthma are always warned about, make sure that you don't feel your heart racing when you're taking the drug and so forth. So there is some overlap in their pharmacology.
Is that to say that there's some organ systems that might be affected more by noradrenaline, less by adrenaline, and vice versa?
There are a few organs where you find only one type of receptor, and the heart is a very good example of that. In heart muscle, you only find a particular type of beta receptor. There's no way of predicting their distribution. But in the brain, again, you find some brain regions have a higher density of one type of receptor than another. But there are very few brain regions that don't express all the receptors. There are, the cerebellum, for instance, has very few receptors of the sort that we call alpha-2. It's mostly beta. The spectrum is enormous.
Doing this podcast, we're oftentimes looking for things that a person could do to optimize their cognition or optimize their health overall. It's sounding to me like the answer for noradrenaline might be that this is really one that we don't want to monkey with our levels. There's not really even a way for us to do so. Is there any way for us to use the knowledge about noradrenaline to optimize our own health, cognition, anything like that?
Well, cognition has different aspects to it. So there's the ability to learn and recall. And noradrenaline doesn't seem to have very much to do with that as a primary process. But what it does seem to be crucial for is attention and arousal and focusing on salient stimuli. And one situation where it seems to be particularly important is to alert the brain when an unpredictable stimulus has happened unexpectedly. That's quite hard to get your head around. But if you know a signal is going to be unpredictable, then you can cope with that. But if suddenly something you think should be predictable isn't, that will give you a jolt. And noradrenaline seems to kick into action in that sort of situation. But it's also terribly important for tuning the activity of a region in the brain, the frontal cortex, that's the clever bit at the front, to make sure that its activity is absolutely right to pay attention to stimuli that we need to watch out for salient stimuli. Now, if the background activity of these neurons is too low, then we're basically on the verge of falling asleep. We're in the lecture, dozing off where we're not taking any notice of the lecture. But if it's too active, like we've had 5 cups of coffee in a row and we're too buzzy, we're then hypervigilant. And certainly if I have too much coffee, I don't know what to worry about first. So getting the activity of those neurons just right for the environment is very important. And Gary Aston-Jones in the States has done some wonderful work showing exactly how that operates. And so noradrenaline activity, noradrenaline release is more like a bell-shaped curve. As you progress from left to right along the bell, then the noradrenaline transmission increases, we get more aroused, we're awake, we're vigilant, we're absolutely spot on to take our exam. We've got just the right amount of arousal going on. But if we go beyond that, then we're in trouble. And that is generally what's thought to happen during anxiety and hyperarousal states and so on. But I have to say the evidence for overactive noradrenergic neurons in anxiety is beginning to creak. But certainly a role for noradrenaline attention is vital. And if we're not paying any attention, then we can't learn anything. There is a chemical element, of course, to that cognition, which relies on noradrenaline, and that involves drugs like amphetamines and cognitive enhancers like modafinil, which interact with dopamine and also noradrenaline, but not much. Methylphenidate, which is used in treating attention deficit hyperactivity disorder. They all modify the actions of noradrenaline and dopamine-containing neurons, and they are all used to influence cognition and attention. So the difference between different drugs of that class is basically whether they target mainly dopamine, in which case they'll have mainly effects on pleasure. It's often called reward. I look at it more as pleasure than reward. Others will target mainly noradrenaline, which is attention, and they both affect movement, whereas drugs that target serotonin is mainly for euphoria and happiness and so forth.
Smart Drug Smarts.
So thank you so very much to Dr. Clare Stanford for taking the time for that interview. This interview almost happened quite a few weeks ago, but the day that we were going to do the interview, she actually broke her foot, which, needless to say, was a bit of a distraction and slowed this down a bit. But we really thank her for making the time and rescheduling. And as a bonus, she's introduced me to one of her peers who's a serotonin expert. So we're going to have a Know Your Neurotransmitters serotonin episode coming up before too long here. We might also be talking with Dr. Stanford again about some work she's been doing recently, where they seem to have developed a very good analog of human ADHD in mice. And there's so much of a crossover between drugs that are used for ADHD treatment and things that wind up being used as general purpose cognitive enhancers, even for people without ADHD, that I can't help but think that would be an interesting and worthwhile conversation to have. So do not be surprised if there is a reappearance of Dr. Stanford sometime in the future here. But for now, let's move on to the ruthless listener retention gimmick.
Smart Drug Smarts. Ruthless listener retention gimmick.
So we've talked about things like synesthesia on this show before, where people can see colors and hear sounds, and where their normal sensory perceptions are a little bit mixed up and kind of cross-pollinated with one another. And I've said that I have synesthete envy. I would love to be able to, at least for a little while, jump into somebody's brain that has some of these unusual perceptions and just see what that's like. Similarly, there are a lot of animals that can sense things that are part of their senses that are completely absent from the human senses. For example, animals ranging from worms to butterflies to sea turtles and wolves are all capable of doing something that humans cannot do, which is sense magnetic fields, in particular the major north to south magnetic fields in the Earth, which help them navigate. You can imagine how if you're a monarch butterfly needing to travel with great accuracy over long distances that you've never traveled before, how something like this could come in handy. But one thing that science has not yet known is how exactly animals are doing this, how they are sensing the magnetic fields. Now, we seem to have an answer to that with a groundbreaking discovery that was recently announced, but not by the right person. Now, everyone involved in this controversy is Chinese, so I'm going to butcher each and every one of their names. But hopefully I will be butchering them all equally, so I will not be playing favorites. But Ji Kan, a biophysicist at Peking University, claims to have discovered a protein that he's calling MAG-R, because it is a magnetoreceptor, and apparently is the physical substrate that these animals use to sense magnetic fields. He started in 2009, he says, using a painstaking whole genome screening to identify a protein that would contain iron and sulfur, and would therefore be a good candidate for a magnetic sensing protein. In December of 2014, he found his molecule, and he started to submit a paper through peer review, getting ready to publish his findings, and in the meantime, behind the scenes, a handful of groups initiated collaborations with him in hopes of using MAG-R as a research tool. And he shared the protein and his related biological materials and data about MAG-R on the condition that these other groups would let him publish his discovery first and that they would include him as a co-author in their papers. He now says, "I never really worried about it because we all agreed that the paper determining the protein should be first." But then controversy struck. On September 14th of this year, 2015, Zhang Shenjia, a neuroscientist at Tsinghua University in Beijing, published a paper in Science Bulletin claiming to use magnetic fields to remotely control neurons and muscle cells in worms by employing a particular magnetism sensing protein, which is in fact the same protein, MAG-R, although the second scientist calls it MAR, not MAG-R, so he's referring to the same compound by a different name. We will link to an article on the Smart Drug Smarts website that goes more into detail about this controversy and the scientific back and forth about who should have been able to publish what in what order. And that in itself is a pretty interesting story, but probably the bigger deal in the grand scheme of things is that scientists now seem to have an answer to this long-standing question of of how it is that certain animals are able to sense magnetic fields and use that as part of their perceptual and navigational system. And if you think this is only modestly interesting and doesn't really matter, I can think of a ton of transgenic organisms that you might be able to make even more useful if you were able to alter their behavior with magnetism. The first thing that comes to mind for me is anytime that we might want to use something like a bacteria or other microorganism to clean up an environmental disaster, but then sort of collect things later, being able to use magnetism as like a homing signal, that just seems like there's a big possibility there. Anyway, really cool technology. The link will be up at smartdrugsmarts.com.
Smart Drug Smarts. We get you thinking.
Okay, you heard it. That is the entire episode. Thanks for hanging around until the very end. If you liked what you heard, we have a bouquet of options of awesome things that you could do to help us out around here at Smart Drug Smarts, including simply coming back next week and listening to next week's episode, because, hey, every little bit counts. But if you're looking for some extra credit and you want to go above and beyond the call of duty, you can head over to iTunes and leave a glowing and positive review for Smart Drug Smarts, which helps us a great deal in their what to recommend algorithm and helps expose us to all new people. Or you could cut out the middleman of iTunes and just tell some friends and colleagues about Smart Drug Smarts. Say, hey, these guys talk about everything from aniracetam to fish to semen. You should really check them out. Or you could head over to axonlabs.io, try out one or both of those stacks, Nexus or Mitogen, and then walk up to people with your cognitively enhanced powers of verbal dexterity and tell them something nice about our little podcast. And last but not least, if you wanted to get really hands-on, you could always recommend something for the Ruthless Listener Retention Gimmick or the This Week in Neuroscience segments. Probably 60 or 70% of those are actually coming from listener recommendations nowadays. By the way, big hat tip to Robert Ness, who turned me on to this week's Ruthless Listener Retention Gimmick. Anyway, that's all for now. I will be back at you next week, same time, same podcast, and with the same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great week, and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.